Alrighty, picking up where we left off. So we had stopped on this slide, and I just want to reiterate this. The key of this slide is the word practical. So yeah, you have to pick a method to destroy what pathogens you actually, need to destroy, and it has to make sense. Okay, so it has to be labor efficient, cost efficient, it has to be safe, and it has to work. So when we talk about killing microbes, let's first define what microbial death is. Now, keep in mind for viruses, this would be inactivation versus death. So it's when various cell or viral structures become dysfunctional, and in the case of a living cell, it has irreversible damage and it can no longer reproduce, even when the conditions are ideal. That's the definition of microbial death. So death begins when a certain threshold of whatever chemical or method you're using is met, and just like the log phase that we saw in microbial growth, where bacteria reproduce exponentially as long as everything is perfect, once you reach a threshold where especially bacteria start to die, they're going to start to die in an exponential or logarithmic manner. Okay, so I want to show you and just kind of go through these four graphs, and these are all common sense, but I just want to show you things in graph form. Okay, so let's start here. So on the left axis, on the y-axis, you see the number of cells that are viable. And what this graph is saying is that, yep, the more cells you start out with, the longer the time it takes to kill all of them and sterilize, in this, whether it's a surgical tool or a liquid. Okay, so common sense. Over here, if it's kind of the same thing, if you have a ton of cells, it takes longer for sterilization to occur. Okay, so you might have to take whatever that is and put it in an autoclave longer than if you didn't have that many cells to start with. Okay, this particular graph talks about vegetative cells versus spores. And you could also include protozoan cysts in this as well. Remember, spores are they have that really hard outer coat instead of just a cell wall. And so they are gonna take much more time to be able to actually get them to burst and destroy them. So yeah, it takes much more time to get rid of spores than it does actual vegetative cells. Okay, and finally, so this refers to microbicidal versus microbistatic. Okay, so if you're using something, and let's say that it's a drug. Okay, so let's say you're taking an antibiotic and you get that antibiotic prescribed to you in microbicidal levels. Okay, awesome. What that means is the cells are going to die. They're going to die exponentially, and then hopefully all of that bacteria is gone that was causing the infection. This is what I had mentioned about how drugs are prescribed. So if the drug is given in microbistatic levels, it prevents further growth, but you're still going to have cells that continue to grow at this level. And this is where we want your immune system to come in and do its job. Okay, and remember, the purpose for that is because there is no such thing as a side effect free drug, and when you increase the concentration, you increase the toxicity to your cells. So if you have a normal functioning immune system, we're going to give you a drug that works in microbistatic concentrations versus microbicidal. So here are some factors that influence how well these chemicals work. Like we mentioned, the number of microorganisms, if you have, a huge number of bacteria versus a small number, that's going to influence the time and the concentration of what you need to use. The nature of microorganisms simply means, what is it? Is it, are they endospores? Are they vegetative bacteria? Are they viruses? Also, the temperature and the pH of the environment, the concentration of whatever chemical you're using. Does it have to be used in, a stock solution, or can you dilute it? The mode of action means how it's actually doing what it's doing, and I'm going to talk about that next. And then finally, the presence of solvents, interfering organic matter, and inhibitors. Okay, talk about that in a little bit as well. So let me cover modes of action. So these are some of the different ways that these chemicals can work. So some of them will actually break apart the cell wall, or they affect the cell membrane. So we've talked about, for example, oh my God, geez, sorry, whoopsie daisy. Okay, boomer. Drugs like penicillin that affect peptidoglycan biosynthesis. There are also disinfectants that basically shred the cell wall. Another target or another chemical, which I want you to know this definition, are surfactants, which attack the cell membrane. Because remember, bacteria, most of them have a cell wall. Every bacteria has a cell membrane. Okay, as do fungi, as do protozoans and worms, etc. So a surfactant is basically a detergent. So every time you do laundry, when you put in that, Tide or whatever you use, you are using a surfactant to break up stains and then to remove, dirt from your clothing. So this is how surfactants work. Here are the little detergent or surfactant molecules. And what they do is they actually intercalate, okay? That's the fancy science word, meaning that they kind of insert themselves into the lipid bilayer of the cell membrane. And if you get enough of those surfactant molecules doing that, the cell membrane just starts to disintegrate. And if you destroy the cell membrane, you're gonna kill that particular cell. You can also remove viral envelopes this way. Okay, then we have chemicals that can bind to stop either translation, transcription, or replication. So we'll talk about this a little more when we talk about drugs in chapter 20, which is coming up next. But there are agents that will actually bind to ribosomes, and if the ribosome can't move, from codon to codon, you're not going to have a protein made, and translation is going to stop. There are also chemicals that can bind irreversibly to DNA. And if you do that, first of all, you don't have replication, right? You also prevent transcription, and if you don't have transcription, you don't have translation. There are also mutagenic agents that actually also intercalate into DNA and cause, the DNA to form kind of clumps, to form what we call thymine dimers, and basically just cause, irreparable damage to the DNA of the organism. So flashback to chapter 2 when we talked about protein denaturation. Okay, so remember, proteins have to have a particular three-dimensional shape to be able to work properly. If you get them to the point where it completely unfolds, like the fried egg example, then you end up with irreversible denaturation, and there's no coming back from this. Okay, the one thing that does this, okay, this is important, is high heat. Okay, cooling does not cause denaturation. The only way that you can denature proteins in terms of heat is to have high heat. There are other things, okay? So, we talked about this. Yeah, if you add a little bit of heat, or if you change the pH a little bit, or, kind of monkey with the salt concentration, yeah, you can cause it to have a slightly different shape. It's gonna work less efficiently. This potentially could renature once that stimulus is removed and then go back to functioning properly. This could not. This is irreversible. Also, where I'm going to show you a couple of examples of heavy metals. So what heavy metals do is they actually go into the, we talked about lock and key mechanism. So for example, they would block the lock, and so the key couldn't fit in. They actually block active sites of various proteins. Okay, so I'm up to 10 minutes. Let me stop here, and then we're going to talk about heat when we come back. Thank you.